My name is Professor Paul Cotter. I'm the Head of Food Biosciences here at Teagasc in Moorepark, and I'm a Principal Investigator with the SFI Research Centre's APC Microbiome Ireland and Vistamilk. Okay, so under the SFI COVID Rapid Response mechanism, we got funding that allowed us to do some fantastic work to do strain typing of the COVID strains that were circulating around Ireland. And that was really a team effort. So what we did was to take advantage here in Teagasc of the expertise and the technology we have through SFI funding of APC Microbiome Ireland and Vistamilk. And we leveraged that to apply it to COVID analysis. But we didn't want to work in isolation. So we worked with a number of different institutes right across the country, universities, but also different clinicians based in hospital, so that we could work as a network rather than a number of different individual labs in isolation.
My name is Fiona Crispie. I have a PhD in molecular microbiology, and I run the DNA sequencing facility here in Moorepark. And at the start of the pandemic, we were approached by a couple of consultants in a couple of different hospitals throughout the country, asking us if we would be interested in, or if we would be willing to sequence COVID isolates as they arose in the hospitals. Obviously, we had no budget for this originally, so when SFI released their early response call, we were really excited and delighted that SFI rose to the challenge, and we applied for the funding under the call. And it enabled us to set up a consortium of sequencing sites throughout the country, similar to the UK Sequencing Consortium, whereby labs throughout the country could do sequencing of COVID isolates. And for this reason, we chose, even though we have quite a large sequencing facility here in Moorepark, partially funded through SFI, we chose a sequencing technology called Oxford Nanopore technology, because they have what's called a MinION, a handheld DNA sequencing device. And these devices are quite reasonable to buy, so a lot of labs have them around the country. Labs that wouldn't have a lot of other sequencing infrastructure. So we chose this technology, and we also have a larger version of the MinION, which enabled us to do a bit more sequencing, or to upscale the amount of sequencing that could be done. And then part of the objectives of the study were, firstly, obviously, to sequence variants as they arose throughout the country. And we've seen that with things like the Delta, the Alpha variant and the Delta variant, how important it is to actually do that kind of sequencing to make sure you keep on top of new variants as they arise. But also it was to kind of encourage and train the hospitals, laboratories that mightn't have this technology already, in how to do this kind of sequencing. So ideally, those kind of labs can do their own sequencing and just be supported by a sequencing core like us, maybe for the bioinformatics or the analysis side of things, or if a lot more sequencing needs to be done, we can step in and help out. So we achieved that. We've trained staff in the hospitals on how to do it, and we've done a lot of sequencing over the last year, and also supported the National Viral Reference Laboratory as they set about doing some national surveillance work. And so I think it's been quite a successful project from that point of view.
I'm Dr. John Kenny. I'm a Senior Research Officer here in Moorepark in Teagasc. And the project, thanks to the rapid response call from Science Foundation Ireland, the aim here was to sequence variants of the SARS-CoV-2 virus that were circulating in Ireland, and to try and build up the infrastructure to keep doing that, to track the progress of the pandemic as it proceeded through the country. We knew from previous work we'd done on different infectious diseases that if you've got a virus that's jumped host, it's probably going to evolve, okay? It's going to adapt for the new host that it's living in. We felt that we had the capability here and nationally to be able to monitor that. Now, keep in mind, this was back in March 2020. We didn't know how things were going to develop. And one of the possibilities was that there would be a vaccine developed, and we didn't know how those mutations could potentially affect the vaccine development. So we thought that in order to be able to track the virus, to track how it was evolving, maybe to track how it was transmitting within Ireland, but also across Europe and into Ireland, that this was an opportunity and a way of being able to do that. One of the aims of all this was kind of capacity building, of being able to build that network, and we were able to achieve that. So, for example, part of what we're doing now, not within the SFI funded work itself, but because we have the methods, because we have the skill set and the protocols all set up, we've actually been able to help with the national overall sequencing capability. So working with Cillian Teagasc and Michael Carr and the group in NVRL, we've been generating data for the national kind of surveillance effort. And that was very much one of the things that we wanted to or hoped to achieve from the project overall. We were able to show, let's say, how the transmission of samples of the virus strains into Ireland was able to take place. You're able to track that on the basis of the genome sequence. And longer term, it gives us a network of people who can work together. So I know, for example, Fiona is working with colleagues in Limerick to develop the sequencing capabilities within the hospital there. And that's something that's going to be in place and be usable long into the future for current work and for the next pandemic.
I'm Vin Maher. I'm a professor of microbiology in UCD. I'm working on two projects that involve close North-South collaboration. One is the Interact Swim project, which was carried out between Northern Ireland and the Republic of Ireland. We aim to develop bathing water quality prediction models, which will of course be beneficial for stakeholders in both jurisdictions. Now, in order to do that, you need to have a methodology and an approach that is comparable. So you can't do two different things in both Ireland and Northern Ireland. So that's why we closely collaborated with our colleagues in AFBI and in Keeping Northern Ireland Beautiful. And as a result, we have developed models that are applicable both in the Republic and in Northern Ireland. The second project focuses on wastewater surveillance, so that means that we are looking at wastewater coming from wastewater treatment plants, and we look for the presence of SARS-CoV-2. So that means that when people get infected, they shed SARS-CoV-2 in their stool, and that can be detected in wastewater. So we collaborate closely with Queen's University, who are carrying out wastewater surveillance in Northern Ireland, and here in UCD we do it for the Republic. So in total we'll be covering around 80% of the total population in the island. Now, it's quite obvious that if you carry out this type of work, that it is beneficial to use the same standards, to closely collaborate, because even though we are two jurisdictions, we are both on the same island. So it is of huge benefit to closely collaborate with our partners in Northern Ireland.
At Queen's University, we're very, very fortunate to have a large number of projects which have a north-south collaboration element. And certainly my group, which is based in the School of Biological Sciences, has benefited from north-south collaborations for at least the last 10 to 15 years. Where we are at the minute is in our research hub looking at SARS-CoV-2 in wastewater. SARS-CoV-2 is shed in feces, both by symptomatic and asymptomatic carriers. So we get a better understanding of the prevalence of SARS-CoV-2 in the community. And then that allows us to direct our public health colleagues to go in and look at specific areas, or informs them what might be happening in individual communities. And as we move forward and we move out of SARS-CoV-2, wastewater surveillance will become that sentinel service where we can see a spike in an individual area, which will allow us then to rapidly respond to that. And the history of the project really started with North-South funding, with funding under the Phase 2 scheme, which was sponsored by Science Foundation Ireland and the Department of Agriculture, Environment and Rural Affairs. And that is where Republic of Ireland programme and ourselves, who were starting the Northern Irish programme, were able to work together and to develop those protocols, develop the ways in which we carry out the testing, the ways in which we can interrogate the data, and the way in which we can inform our public health colleagues. So at the minute, we are currently surveying approximately 50% of the population in Northern Ireland. And through extended funding from the Department of Health, from the Public Health Agency, from the Department for Infrastructure, from the Northern Ireland Environment Agency, and the Department of Agriculture, Environment and Rural Affairs, as well as our colleagues in NI Water, we will be extending that to approximately 70% of the population in the coming months. Vim, on his side, is measuring around 80% of the population. So we have, across our shared island, this ability to monitor over 70% of the population just by looking at their wastewater, and that is a very powerful public health tool. The other part of my collaboration is on waste, and that's where we have the link to wastewater. So for many years I've been working with Professor Vincent O'Flaherty in NUI Galway, and we have had multiple Irish Environmental Protection Agency grants from Science Foundation Ireland and our own Department of Infrastructure, and also now an Intertrade Ireland grant. And currently we're developing what we call the Irish Nutrient Sustainability Platform. And that is to bring business together from Northern Ireland and from the Republic of Ireland, and to link them up with research performing organisations to develop new value streams. to provide a safe space where they can talk about the issues that they have with waste with the regulators on both sides of the border. So as we move forward, as we see SARS-CoV-2 levels going down, Vim and I are now starting to think, well, what other things could we look at in our wastewater? Could we look at other pathogens, other human pathogens, other animal pathogens, antimicrobial resistance, other chemical markers? And under our Shared Island Initiative, we now have this capacity through these North-South funding programs that have been developed and are being developed to really make an impact upon the health of the island. So providing our agencies, our Department of Health, our environment agencies, our wastewater companies, our public health agencies across both sides of the border with vital information as to what's happening in our communities across the island.